Good afternoon, everyone. Today I will be talking about chromatin interaction networks, revealing unique connectivity patterns for super enhancers and broad domains in 3D chromatin, which was recently published in Scientific Reports. So, as we know, we have different regulatory elements such as promoters and enhancers, and they interact with each other with these chromatin interactions. Leading to our first research question is whether or not different chromatin interactions have different connectivity patterns, whether or not these different regulatory elements have different connectivity patterns between different regulatory elements. Furthermore, we know that there are cell-specific regulatory elements for promoters and enhancers known as broad domains and super enhancers, leading us to our second research question of whether or not there are differences in interaction patterns between cell-specific regulatory elements and their non-specific ones. And finally, we ask, can we use these differences to infer, for example, cell specificity between these elements? So in order to answer these questions, we chose to use chromatin interaction networks, where we first build our chromatin interaction networks by integrating chromatin interaction data such, with either chia-PET, high-C, or high-CHIP data, with DNA-seq data, which annotates accessible chromatin, where the accessible chromatin data will define the nodes in our networks, and the chromatin interaction data provides the edges within our networks. Next, we annotate our networks by first using ChromaHMM states to annotate the different regulatory elements. And then next, we annotated the cell-specific promoters with broad domains and cell-specific enhancers with super enhancer states. With our annotated chromatin interaction network, we then use various network mining and machine learning algorithms to process our interaction networks. So here's an example in the MIC locus where we have our different tracks used for building our chromatin interaction networks. For example, we have our RNA pull-2 chia-PET data, DNA-seq data, ChromaHMM states, broad domains, and super enhancers. And we use our previously published tool Quinn, which is available at quinn.jax.org, to build our annotated chromatin interaction network. And so some of the things that some of the measures that we use for analyzing these networks include global connectivity measures, which include connectivity degree, closest harmonic, and between a centrality, and local connectivity measures, which use graphlets, which were first introduced by Natasha Przulich in 2007. We discussed one of these measures, actually the simplest one, connectivity degree, which is defined as simply the number of edges connected to a node. And what we see is that among the different regulatory elements, promoters, broad domains, and super enhancers have a higher connectivity degree than non-specific enhancers and other regulatory elements defined by ChromaHMM. And in particular, between promoters, broad domains had a even higher connectivity degree than typical promoters. And below, you see an example in CF7 and in K562, where we have our gene EMP2, which is a broad domain, which has a higher connectivity with other regulatory elements. In particular, these regulatory elements are super enhancers. And then in the same gene in K562, we see a typical promoter, which has a very low connectivity comparatively. So next, we want to analyze whether or not we could identify whether or not a promoter or enhancer would be cell-specific. And to do this, we created feature matrices including network-related features and genomic-related features, and input this into a support vector machine model. And we evaluated our results using the receiver operating characteristic, the area under this area under the curve for this characteristic, where a value of 1 will be a perfect predictor. And what we notice is that in the case of super enhancers versus typical enhancers, using this data ended up being highly predictive with an area on the curve of 0.84. And furthermore, we also noticed that network-related features were the most predictive. And then when we looked at the performance across different cell lines as well as different assays, including high CHIP and high C, we noticed the same pattern where the network connectivity features were very indicative of whether or not an enhancer would be a super enhancer. So in conclusion, we built chromatin interactions networks in 3 cell types and in 3 different chromatin interaction assays. Identified network connectivity patterns specific to super enhancers in broad domains and established that these patterns are discriminative for cell-specific activity using support vector machine models. I'd like to acknowledge the co-authors on our paper, Dugu, who's my current PI at JaxGM, as well as Eladio. Also Paula from UConn Health and my PhD advisor, Dr. Dongguk Shin at UConn. You can read more about our research in our scientific reports paper. You can find our tool for building chromatin interaction networks at quinn.jax.org. And if you have any more questions, my poster ID is A63. And we presented that tonight. All right, thank you.